Okay, this is Nina Lebrun doing my Synaptic Transmission video for Dr. Cortez's Neurobio class for fall 2018. So what I've drawn here is the presynaptic neuron, and then a little synapse here and a postsynaptic neuron over on the right. These little red things are the vesicles that I've drawn, and then inside them in the green is the neurotransmitters. So what I'm going to start with is just kind of the basic mechanism behind synaptic transmission. What happens is, in the last videos I discussed the mechanism for how a neuron sends an action potential. Basically, now we're at the very end of that action potential, so we're at the axon terminals, and once that action potential reaches all the way down here, the change in voltage that it brings is going to cause a whole big influx of calcium to come into this into the presynaptic neuron, and it's going to bring the voltage way up. But what it's also going to do is these calcium ions are kind of going to chaperone the vesicles over to the cell membrane. And what's unique about these is the membrane of the vesicles is made out of the exact same the bilayer as the cell membrane itself. So what it's going to do is it's going to fuse like this one has down here. The two membranes will just fuse with one another. And when it fuses, it's going to kind of spill its guts. It's going to release all of its neurotransmitters into the synaptic cleft. And what I've drawn in this one, what I'm talking about is a chemical synapse. There are electrical synapses, which are really, really itty-bitty and have gap junctions in them. But this is a chemical one, which is somewhere between 20 and 50 nanometers wide, which is really, really itty-bitty. But it helps because now these cells are so close to one another, it can release just a little bit of neurotransmitter from this one. And on the postsynaptic neuron, we have these different kinds of receptors that are going to receive that neurotransmitter, hopefully. And so this vesicle is only going to release one kind of neurotransmitter, but that one neurotransmitter might work with several different postsynaptic receptors. So it might have a G protein coupled receptor. Those are going to tend to be a little bit slower because what happens is it'll receive the neurotransmitter, and then what it'll actually do is activate a G protein on the inside, which goes on to activate a secondary pathway. So it's going to be a little bit slower than an ion channel receptor, which is very direct. Like this neurotransmitter is going to bind, and it might let in sodium, for instance, which is going to create an excitatory postsynaptic potential because it's going to be depolarizing this postsynaptic cell really quickly. So an ion channel that's excitatory might let in something like sodium, versus if it lets in chloride, that's going to be inhibitory. It's going to hyperpolarize that cell. So depending on what receptors are present on this postsynaptic neuron, the same neurotransmitter from the presynaptic might create either an inhibitory or an excitatory postsynaptic potential. And then, like I talked about in the action potential video, you're going to need to sum up either by location or by time all those different potentials to either create or not create an action potential in the end in this postsynaptic neuron. So anyways, once you've got all your neurotransmitters in the synaptic cleft, you got to bring them back into the presynaptic cell because they don't go ever into the postsynaptic cell. They just get kind of recycled. So you might have a glial cell out here that absorbs all those different neurotransmitters and breaks them down and then sends the pieces back to the presynaptic neuron. You might just have enzymes in the synapse that break them down and again take the little building blocks and send them back in where they can be repackaged into vesicles. You might just have a more direct mode of reuptake in this presynaptic neuron, where it just takes the neurotransmitters right out of the synapse back in and packages them into the vesicles. Yeah, so that's how you're going to get them out of the synaptic cleft. Basically, you probably have some autoreceptors on this presynaptic neuron that the neurotransmitter might bind to and say, hey, look, we've put enough neurotransmitter into the synaptic cleft, you can stop now. And then after that, it's going to kind of signal it to reuptake in one way or another, either by the enzymes, by directly reuptaking them, or by taking them from a glial cell that's kind of picked up the extra. The last thing I'm going to talk about is the different classes of neurotransmitters. So there's 4 different classes. The first one is going to be amino acids. These are super, super common. These are going to be things like GABA, glycine, glutamate, They're made out of amino acids, just, like what proteins are made out of. The other one is monoamines. This is going to be the catecholamines and indolamines. Those are like dopamine, epinephrine, norepinephrine, serotonin is one of the indolamines. Next, you're going to have neuropeptides. These are things like endorphins, which are much, much bigger than the other ones. And then acetylcholine sometimes is grouped just into its own little group. It just acts everywhere, especially at the neuromuscular junction. That's a really common one. And in one of my classes, I saw it grouped into its own category. So those are the categories of neurotransmitters. Again, each of those neurotransmitters might have multiple different kinds of receptors that can either create an inhibitory or an excitatory postsynaptic potential, depending on the receptor, not just on the neurotransmitter from the presynaptic neuron. So that's how synaptic transmission works.